Welcome to our dilution video. First up is Mr. Logan Montgomery. So first, we will be talking about the meniscus in a tube. The meniscus is the curve in the upper surface of a liquid close to the surface of a container, which is usually caused by surface tension. It can be either concave up or convex. It all depends on the liquid and the surface. The meniscus occurs because when liquid water is confined in a tube, its surface has a concave shape because water wets the surface and creeps up the side. To read the meniscus in the tube, look towards the bottom of the meniscus where it is concave and read it there. In this video, the meniscus is properly read and recorded in this lab notebook. Next up is Brendan Moore. We will be talking about stock solutions and dilutions and the difference between the two. A stock solution is a concentrated solution that is pre-made and will be diluted to some lower concentration for actual use in experiments, such as we did here. Dilution is the process of reducing the concentration of a solute in solution, usually simply by mixing with more solvent. In this case, the solvent is water, and we will be adding more water to a smaller amount of the blue stock solution to dilute the stock solution and use it for our experiments. Please note the solution is diluted in the volumetric flask using a transfer pipette. Also, please note the solution shown here is drawn into the pipette, not into the bulb. The bulb simply helps to draw the solution in. And as you can see here, the meniscus is at 0, which means that there are 10 milliliters to be exact in this pipette that we are transferring into the volumetric flask. After we are finished pouring the solutions in, the meniscus in the volumetric flask sits right at the mark. In this section of the video, a solution of a weaker concentration is made using a larger quantity of water and a smaller quantity of the blue liquid. As you can see here, the solution is released from the transfer pipette by releasing the thumb from the top of the transfer pipette. And again, solution is drawn into the pipette using the blue bulb. Now the solution is being added to the Erlenmeyer flask of just water. There's some trouble here, but it is all worthwhile as the meniscus sits right at the mark of the desired value. After the water and the solutions are poured in, the meniscus in the volumetric flask sits right on the mark. At the end of the laboratory space is properly cleaned and excess reagent is placed into the proper waste containers.